Okay, good afternoon. I'm very happy to open this session that looks like an exciting session on different aspects of stemness, signaling, stromal cells, and more. And we will begin the session by the presentation by Ren Xu on a novel function of collagen hydroxylation in regulating the HIF pathway. Please, Ren.
Well, first, I want to thank the committee for inviting me to give this talk. And also, I appreciate they put my talk in this section, not in the morning section, because I don't need to get up, at midnight. So now it's 7:30 at East Coast time. I'm not sure, should I say good morning, everyone, or good afternoon, everyone? But anyway, research in my lab mainly focus on ECM function in breast cancer progression and metastasis. So as many of you know, ECM extracellular matrix remodeling is crucial for cancer development and progression. So there are many ECM proteins have been identified in the tumor microenvironment. For example, the basement membrane protein, laminin, collagen, nitrogen, perlecan, and the interstitial ECM protein, for example, fibrillar collagen, fibronectin, and decotin. And there are about hundreds ECM and ECM-related genes have been identified. And during the tumor progression, those ECM proteins, is very important for the cell proliferation, cell adhesion, migration, invasion. They're involved in many different cellular process. So when I start my own lab, I try to look at the ECM remodeling at the whole genome level. Therefore, we identified like 700 ECM genes and ECM-related genes, and then we collect their expression profile from more than 500 human breast cancer tissue. And then we run this called transcription network analysis, identified this ECM network in the human breast cancer tissue. All those ECM genes, their expression are significantly correlated. And we also found the malignant tissue, the activation or the expression of these ECM network genes are significantly higher than the normal tissue. We have identified a number of ECM, a number of harbs from these ECM networks. One of the harb is called P4H. P4H is a collagen proline hydroxylation enzyme. It can induce proline hydroxylation on a collagenous region, triple collagenous region. This hydroxylation is very important for the collagen to form triple helix region. And therefore the protein can be folded correctly and secreted from this collagen fiber in the tumor microenvironment. So P4H is a tetramer. It has two alpha subunits called P4HA and two beta subunits called P4HB. So there are three P4HA subunits has been identified from 1 to 3. So actually P4HA is the major isoform in most cells. It accounts about 60 to 70% activity of the P4H activity. Knockout this gene can lead to the embryonic lethal, indicating the function of this gene is very important. So since we identified P4HA as a harb of this ECM network gene, we want to know expression pattern of this gene in breast cancer. So we performed the tissue microarray analysis. And from this slide, you can see P4HA expression is significantly higher in triple negative breast cancer. Triple negative breast cancer does not have ER, HER2, PR expression. That's why it's called triple negative. And therefore, there's no targeted therapy available for this type of cancer. So most common therapy for triple negative breast cancer is chemotherapy. However, 50% of the triple negative breast cancer eventually will develop a resistance to chemotherapy. So therefore, this disease is very aggressive and very difficult to deal with. So we identify P4HA is highly expressed in triple negative breast cancer. And here is the quantification data. You can see the triple negative breast cancer have much higher expression of P4HA1. And in addition, you can see from stage 1 and stage 3, the P4HA positive rate is significantly increased, suggesting the increased P4HA1 expression may also contribute to the tumor progression. And we also analysis the expression in a panel of breast cancer cell line. You can see the triple negative breast cancer cell line have significantly higher expression of P4HA1. So then we want to know, is the increased expression or high expression of P4HA1 functionally important for triple negative breast cancer progression? So we have using shRNA to knock down P4HA expression in metastatic breast cancer cell line, MDA, MD231 cells. And then we inject those cells into the SCID mice from tail vein. You can see the control mice develop lots of metastasis, while knockdown P4HA1 can dramatically reduce the metastasis lesion in the lung and in other tissue and organ. And at the same time, Dr. Simenza's lab, they reported P4H gene is very important for tumor metastasis. So then the next question is, how P4HA1 contribute to the triple negative breast cancer progression? So to answer this question, we actually did a screening, identified like 100 of P4HA1 co-expressed gene. And then we performed gene ontology analysis. We find, the gene co-expressed with P4HA1 mainly contribute to the response to hypoxia, response to glycolysis, and glucose metabolism. Once we identify those pathways is related to P4HA1, one pathway come to my mind is the HIF1 pathway, because the HIF1 pathway can regulate the response to hypoxia, can contribute to induce the glycolysis. At the same time, HIF1 pathway is also very important for angiogenesis and cancer progression. That has been well studied. And there are many also studies showing that HIF1 pathway involved in the stemness and drug resistance during the cancer progression. So then we further analysis the way the P4HA1 expression is associated with the activation of HIF1 pathway in human breast cancer tissue. Here we use the HIF1 gene signature as an indicator of HIF1 pathway activation. You can see the high expression of P4HA1 is associated with activation of the HIF1 pathway in cancer tissue. So to determine whether P4HA1 can really regulate HIF1 pathway, we first overexpress P4HA1 and P4HB in non-malignant mammary epithelial cells. Those cells usually have low P4HA1 expression, and you can see P4HA1 overexpression can significantly increase HIF1 alpha protein level in both normoxia and hypoxia conditions. So we also examine the HIF-1 alpha protein level in the P4HA1 silenced breast cancer cells. You can see silenced P4HA1 can reduce HIF-1 alpha protein level. And there are a number of P4HA1 inhibitors have been identified. One for DPCA and DHB are two of them. And you can see treatment with these two inhibitors can also reduce the HIF-1 alpha protein level in breast cancer cells. To further determine whether P4HA1 can regulate HIF-1 alpha activity, we performed a HIF-1 alpha luciferase reporter assay. This reporter plasmid containing the HIF-1 alpha beta binding site. Therefore, the luciferase activity can be used as an indicator for the HIF-1 alpha transcription factor activity. And you can see silenced P4HA1 can significantly reduce the luciferase report activity, suggesting the HIF-1 pathway is also inhibited by silenced P4HA1 or treatment with P4HA1 inhibitor. So our data clearly show P4HA1 is an important regulator of the HIF-1 pathway. Then the next question is, how P4HA1 regulate HIF-1 alpha? So regulation of HIF-1 alpha can be achieved at different levels. The first is at the transcription level. We can measure the HIF-1 alpha mRNA. And the other one is translation and protein stability level. So we first check whether overexpression or silence P4HA1 can regulate HIF-1 alpha gene expression. And we didn't see a significant change of HIF-1 alpha mRNA level in the knockdown or overexpression cells, indicating the regulation is not at this transcriptional level. Okay, then we focus on the protein. So the first, we look at the protein stability. So here is the Western blot showing the HIF-1 alpha protein stability in control cells and also in the silenced cells. You can see silenced P4HA1 can significantly increase the HIF-1 alpha protein degradation. And here is the quantified protein degradation curve. You can see this is the control, and this is the P4HA1 silenced cells. And in addition, we also found the ubiquitination of the HIF-1 alpha is dramatically increased in the P4HA1 silenced cells. All those data indicating the silenced P4HA1 can enhance HIF-1 alpha ubiquitination and protein degradation. So, 2 or 3 years ago, the Nobel Prize have been given to the identification of the HIF-1 and HIF-1 regulation mechanism. So it is well established, and probably all of you know the HIF-1 alpha degradation is regulated by oxygen level through the proline hydroxylation. So this reaction is catalyzed by PHD, is a protein hydroxylation enzyme. The reaction required oxygen and alpha Kg and will generate CO2 and succinate. So from this reaction, you can see at a high level of oxygen, and the protein will be ubiquitinized and degraded. And meanwhile, if alpha Kg level remain high, it will also increase this reaction, induce the HIF-1 alpha protein degradation. So we first wonder whether the P4HA1 expression can regulate PHD, the key enzyme induced HIF-1 alpha hydroxylation. Unfortunately, we didn't see any significant change in the PHD 1, 2, 3 level in P4HA silenced cells, indicating the regulation is not at the PhD. So then we think about, the P4HA1 is a collagen polyhydroxylation. If you look at these two hydroxylation, collagen hydroxylation and HIF1 hydroxylation, they both use alpha Kg and oxygen as a substrate and generate succinate CO2 as a byproduct. So, it seems like these two enzymes share the same substrate, generate the same byproduct. So we then wonder whether the increased P4HA1 level can inhibit the P4HD activity, by modulating those metabolites levels. And when we have this hypothesis, we go back to the literature and look at the enzyme activity of the HIF1 hydroxylation enzyme and the collagen hydroxylation enzyme. And you can see the Km for the collagen hydroxylation, Km for the oxygen, is much lower in the collagen hydroxylation enzyme, which means the collagen hydroxylation have much high affinity for the alpha Kg, and they're probably a very good competitor for the alpha Kg with P4HD. So to test this hypothesis, we performed a stable isotope resolved metabolomics analysis. It's called SERM analysis. Basically, we use the stable isotope label the glucose and to feed the cancer cells. And then we can see silence of P4HA1 expression in those cancer cells can significantly increase the alpha Kg level, suggesting the P4HA1 is a very important generator, consumer of the alpha Kg in cancer cells. So then we further analysis use alpha Kg quantification assay. You can see silence P4HA1 can dramatically increase the alpha Kg level in cancer cells, while overexpression P4HA1 can reduce alpha Kg level. So this further confirmed our hypothesis. P4HA1 may inhibit HIF1 alpha hydroxylation and degradation by reducing alpha Kg level. So to further test this hypothesis, we performed a rescue experiment. Since we see the alpha Kg level is downregulated in P4HA1 overexpression cells, so we add alpha Kg back in the overexpression cells, and then to see whether we can rescue the HIF1 alpha protein degradation. And from here, you can see in the control cells, overexpression can induce the alpha Kg, can reduce alpha Kg level, further enhance HIF1 alpha protein stability and protein level. However, once we treat those cells with alpha Kg, the P4HA1 overexpression have little effect on the HIF1 alpha protein level. So we further use the luciferase assay, confirmed alpha Kg can inhibit P4HA1 induced HIF1 alpha pathway activation. Well, it is well established the HIF1 pathway is a key regulator for cancer cell or cancer stem cell renewal. HIF1 alpha can enhance the glycolysis in the stem cells, reduce the oxidative phosphorylation. So those changes in the cell metabolism is crucial for the self-renewal and multipotency of cancer cells. Those pathways have been well established and well documented, so I will not talk too much about it. So since we identify P4H1 is an important regulator of the HIF1 pathway, we ask whether P4H1 contribute to the cancer cell stemness. So we first examine the P4H1 expression in cancer cell population and the tumor cell initiate cancer stem cell enriched cell population. Here we use CD44 positive, CD44 high, CD24 low cell population as a tumor initiation cell population shown at here. So we have a number of cell clones isolated from human mammary epithelial cells. Some of them have high level of the tumor initiation cells, like a clone 3. And this one is just a regular mammary epithelial cells, okay? And we found that P4H1 expression is significantly higher in the tumor initiation cell enriched cell population. And also you can see the E-cadherin level, HIF1 alpha level also changed in those tumor initiation cell population. And tumor sphere have been used to enrich tumor initiation cells or cancer stem cells. And we also find in the tumor sphere formed by human breast cancer, primary human breast cancer cells have significantly higher expression of P4H1 compared to the primary tumor. So this data showing that P4H1 expression is highly expressed in cancer stem cells or tumor initiation cells. So then we want to know whether silence P4H1 expression can reduce the tumor initiation cell population. We performed 2 assays. Firstly, the tumor sphere assay. You can see silence P4H1 can significantly reduce the tumor sphere formation, also reduce LDH high cell population. So LDH high has been used as a marker for cancer stem cells in breast cancer. So to further determine whether P4H1 regulate cancer cell stemness through HIF1 pathway, we performed a rescue experiment by overexpression HIF1 alpha. Here we use the HIF1 alpha mutation called PPAA mutation. So this PPAA mutation mutated the proline site in the HIF1 alpha to alanine. So therefore, this protein stability is not further regulated by the hydroxylation, no matter whether you culture the cells in the low oxygen, high level oxygen, or high level alpha kg, low level alpha kg, this protein will remain stable, okay? So therefore we can use this construct to rescue the HIF1 alpha protein expression in the P4H1 silenced cells. You can see once we restore HIF1 alpha activity in the silenced cells, the tumor sphere formation activity also restored, and LDH high cell population increased. This data clearly indicate the P4H1 regulate cancer stemness through the HIF1 pathway. So we know that cancer stem cell is a driver for cancer progression, including cancer metastasis and drug resistance. During the chemotherapy, even though majority of the cancer cells can be killed by chemotherapeutic agent, because of those tumor initiation cells, they are proliferate very slow, and they have high expression of many enzymes involved in drug resistance. So they usually cannot be killed during the chemotherapy. And once you remove the chemotherapy agent, those cancer stem cells can cause the cancer recurrence either in the local or at a distance place. So since we see the P4HA expression is very important for the cancer stemness, so we wondered whether the P4HA expression also contributes to the chemo resistance. So here is just the Kaplan-Meier analysis of the P4HL1 expression association with triple negative breast cancer patient survival and chemo resistance. You can see in the triple negative breast cancer patient, high expression of P4HL1 is associated with poor prognosis. So this trend is even obvious in the patient who received the chemotherapy. You can see that for the chemotherapy treated patient, high expression associated with poor prognosis, short recurrent-free survival. This data suggests P4HL1 expression may contribute to the drug resistance, chemo resistance in the triple negative breast cancer. So to test this hypothesis, we silenced P4HL1 expression in triple negative breast cancer, and then treat the cells in the xenograft model with chemotherapy. And you can see silence of P4HL1 itself have little effect on the triple negative breast cancer cell tumor growth without any treatment, okay? However, once you treat those xenograft with docetaxel, you can see the docetaxel can slow down the tumor growth in the control cells, while in the P4HL1 silenced tumor cells, docetaxel treatment can induce the tumor regression. We treat those tumors with chemotherapy agent for like 2 or 3 weeks and then withdraw the chemotherapy agent. You can see the tumor remain at very small volume for a while, but eventually start to grow, indicating, you know, even silence P4HL1 at certain level still cannot fully induce the tumor remission. Here is just some proliferation assay and cell apoptosis assay in the tumor tissue. Since we have a couple of P4HL inhibitors available, you know, none of those inhibitors are FDA-approved drugs. You know, they're just some chemicals have been used in animals. So we know that we can use this chemical for the mouse experiment. So we did the single treatment and the combined treatment with these inhibitors and the chemotherapy drugs, and we find combined treatment with the P4HL inhibitor and docetaxel can significantly delay or inhibit tumor growth in the xenograft model. So eventually we want to target P4HL1, you know, in human breast cancer patient. But one challenge right now remains is the P4HL inhibitors identified have not been used in any clinical trial. So we don't know whether they are good drugs or bad drugs for human, even though it seems like it works in the mouse model. So therefore, we think maybe we can develop a high throughput screening assay to identify P4HL inhibitors from FDA-approved drug library. You know, once we identify an inhibitor from this drug library, we can then easily initiate a clinical trial, use those chemicals because those are all FDA-approved. Their safety have been demonstrated in other disease. So how to develop this high throughput assay? You know, you probably still remember I just mentioned to you the collagen hydroxylation reaction required alpha Kg oxygen and will generate succinate and hydroxylated collagen protein. So therefore, there are 2 ways to do the screening. First, we can, you know, use hydroxylated substrate like GPPG, develop a colorimetric assay to measure the hydroxylated GPPG. Another way is we can measure the other product of the reaction called succinate. We can use hydroxylase assay, succinate hydroxylase assay to measure this substrate. So we have expressed the P4H complex using the 293 cells and get this protein purified. You can see this protein form the tetramer after purification. And then when we test the hydroxyproline colorimetric assay, you can see we can use this assay to measure this reaction, but this reaction is not very sensitive. You know, when we change the substrate concentration, the enzyme concentration, the enzyme activity doesn't change that much. However, the succinate glow assay seems to me is quite sensitive. So therefore, we decided to use this assay to do the screening. And then we screened like 1,400 FDA-approved chemicals use this assay. We identified about 2,000 of them can significantly reduce P4HA activity. And we choose 2 of them to further determine their activity, inhibitory activity in the assay. And you can see the silodosin and the tiglopitin, these 2 chemicals can inhibit P4HA activity in a dose-dependent manner. However, when we remove the P4HA enzyme or use the P4HA enzyme inactivate mutant, this silodosin or tiglopitin cannot further inhibit this reaction, indicating the inhibitory activity is specifically for the polyhydroxylation enzyme activity. And then we use a tissue culture model to further showing that treatment with these 2 chemicals can reduce the collagen secretion in cancer cells, and the HIF1 activity is also inhibited by this inhibitor. So those data show, we also performed the 3-dimensional culture assay to see whether these 2 inhibitors can inhibit tumor progression. We use MDA231 cells. You know, these cells is very aggressive in the 3-dimensional culture. You know, they form these aggressive branching structures. When we treated with these inhibitors, you can see the tumor invasion was dramatically reduced. So here is just the quantified result. So far, you know, we have identified a number of FDA-approved drugs that can inhibit P4HA activity. We show that P4HA activity is very important for chemoresistance. So next, we will test whether those inhibitors can inhibit tumor progression in vivo in the xenograft model, sensitize the tumor cells to chemotherapy. And to summarize, we have identified P4H as a hub of the ECM network, and it highly expressed in the triple-negative breast cancer. And we show the P4H have a crosstalk with PhD in the HIF1 alpha hydroxylation pathway. Increase the P4HOA1 activity can reduce alpha Kg level, therefore inhibit HIF1 hydroxylation, induced HIF1 alpha stability, and increase HIF1 alpha protein level. Further induce the cancer cell stimulus, increase cancer metastasis and the chemo resistance. So at the end, I want to thank the people who performed the research, like Dr. Gaofeng Xiong and Dr. Shike Wang, and also the students and other members in my lab, my collaborators at the UK, at Lawrence Berkeley National Lab, in Bissau, and Kyoto University, Dr. Nagata. He gave us lots of help with mouse experiment and also the funding support. Thank you for your attention. I'm happy to answer your question.
Thank you. Thank you very much. And I would like to ask if there are questions from the audience.
Mm-hmm.
Yes.
I would like to ask a question. First of all, Dr. Xu, excellent talk. Thank you very much. It was very, very interesting. I have a question as far as when you inhibit the inhibition you did with P4HA1 with shRNAs or with the compound you used in vivo. In vitro, you see that inhibition affects sphere formation, etc. But in vivo, you don't see much of an effect when you just inhibit P4HA1 alone. How do you reconcile this difference, the difference in vivo and in vitro?
You mean the knockdown experiment and the...
Yeah, so here, for example, you don't really see an effect with DHB
Exactly.
and then with the shRNA either, but in vitro, when you do knock down or inhibit, you see an effect of, for example, in sphere formation.
Yeah, the tumor sphere formation is mainly to check the, I would say, the cancer stem cell self-renewal. It's not really focusing on the tumor proliferation or whatever. When we look at the Cas67 staining in the tissue culture, you know, the DHB, which is a P4HA inhibitor, they still can slightly reduce the tumor growth. They can inhibit Cas67 positive rate a little bit. But silence of P4HA1 have little effect on the tumor growth and cell proliferation. You know, the difference between the gene silence and DHB, you know, may rely on the specificity of this inhibitor. For example, the DHB can target not only P4HA1, it can also inhibit the P4HA2, P4HA3. You know, all the P4HA isoforms can be inhibited by this inhibitor.
Did you include the DHB in your high throughput analysis?
Yes, we did. And
did it perform better or worse than the two compounds you identified?
So we use it as a threshold. You know, only the inhibitors have more potency than this one. You know, we pick them up.
Okay.
So basically, all the inhibitor we identified is more potent than this one. Okay. So the tumor sphere is a I would say it mainly showing the tumor stem cell or the tumor initiation cell self-renewal, and it's not just measure the cell proliferation. Yeah.
All right, thank you very much.
Thank you.
Hi, can I also ask something?
Sure.
You actually showed that P4H regulates both HIF1 alpha and stemness. And I wanted to ask if cancer stem cells that do not express it, P4H, will they have a higher sensitivity to hypoxia conditions?
Well, before we do that experiment, we actually examine the P4HA1 expression in the tumor initiation cell enriched population. We clearly can see the tumor initiation cell have high level of P4HA1 expression, also high level of HIF1 alpha. So, you know, according to the literature, the high expression of P4HA1 alpha or the HIF2 is very crucial for the tumor initiation cells or the stem cell self-renewal because they can survive in the low oxygen level. You know, many of the stem cells, you know, are in the bone marrow. You know, that oxygen level is very low. And in those low oxygen level, they need to shift to the glycolysis rather than completely rely on the oxidative phosphorylation. So therefore, the HIF1 activation or the HIF2 activation is very crucial.
Okay, thank you.